Hi, everyone. Thank you. Thank you for the invitation to Pharma 2022. I'm Dr. Luis Jesús Villarreal Gómez. I'm a research professor from the University Autonomous of Baja California in Tijuana, Baja California, Mexico. And today we will expose the topic antimicrobial electrospun nanofibers for wound dressings. First of all, I'm from Mexico and I live in Tijuana, Baja California. It's a city that is at the border of United States of San Diego border. So I put some pictures about my institution, some photographies. As you can see, is in the middle of nowhere. There is a lot of nature around the university where I work. And but uh, like for example, in this month, in March and February, the the view is beautiful because we have a lot of flowers, a lot of green green mountains. Where I will speak, I will give you some introduction about the topic that I will share with you today. I will talk about our previous work. I will explain the electrospinning technique that is a technique that in our lab is the expertise. I will talk about bioactivity of silver nanoparticles, some methods for incorporation of these silver nanoparticles on fibers, some preliminary results that we have already, and some future work and some preliminary conclusion because this project is still in process. Well, there is a lot of literature that has proved that antimicrobial activity of silver nanoparticles. There is pretty interesting how these silver nanoparticles can be presented in several uh, modalities, in several kind of products, in several versions. And now with us, we will present to you the silver nanoparticles loaded in electrospun fibers. So these electrospun fibers with these silver nanoparticles together, these two technologies creates some membranes that has antimicrobial properties and can be used for food packaging, some face mask filters, wound dressings, some dental implants, and so on. We have been published some work before about the use of silver nanoparticles in electrospun fibers. I will explain later what is an electrospun fiber, but we have been working since 2020 in this project. We prepare we propose a wound dressings with silver nanoparticles in electrospun fibers, and also we make a review. We compare different methods for silver incorporation of these nanofibers. This article was theoretical, and right now we are doing the work in the lab experimentally. So, what is the electrospinning technique? Where the electrospinning technique is a method that the main the main objective of this technique is to produce or fabricate nanofibers. As you can see in the background of this slide, you can see some fibers that are in a nanometric scale. These nanofibers can be loaded with different bioactive materials like proteins, vitamins, some natural molecules, drugs, uh, plasmids. We can modify the surface of these nanofibers, or we can load in the inside, in the core, we can load several molecules. And as you can see, we create a three-dimensional structure, something like this, with this technique. So first of all, we prepare a polymeric solution. This solution has to be conducted. We load it in a shrink and we put it in a shrink pump. Then we 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 put in this shrink pump, we put, or we program a certain uh, release rate. So then we have a metallic collector, usually aluminum, but we can use another kind of metals. But as this is a biomedical application, so we usually use metals that has no interference in when we talk about immunological responses. Then we have a metallic collector, and we put a high voltage supply with electrodes. We put we locate one electrode in the metal collectors and the other electrode to the shrink tip. We put it there, and when we start releasing the polymeric solution, we put on the high voltage supply, usually 5 kilovolts or 20 kilovolts. We apply the voltage, and then it's created an electrical forces that attract the polymeric solution to the metallic collector. So um, the surface tension is broken, and then uh, a little fiber is created in the air, and the solvent is evaporated. So it solidifies in the air, creating or forming the fiber in the air, and it's collected randomly in the collector. Why electrospun nanofibers are interesting? Well, you can see the picture at the right, and you can see a randomly distribution of these nanofibers. And for tissue engineering, we simulate extracellular matrix in order to promote the proliferation of cells. So cells get inside this three-dimensional structure and proliferate faster. These fibers are in the range of 100 to 1,000 nanometers. There are several publications that reports some sizes around 10 nanometers or less. These structures are very interesting because they have a high surface area. So this means a higher contact sites in a minor spatial radius. And this high surface area can be controllable. Also, we can obtain a membrane we a small pore size, but this pore size also can be variable depending on the condition that I will explain later. These membranes of these fibers has a low density. You can use biocompatible polymers, biodegradable depending on the application. They can be flexible, elastic, with desired mechanical properties, and also depending of the loading, you can create bioactive nanofibers, for example, or fibers that are loaded with silver nanoparticles. So they had the antimicrobial bioactivity. I can here in this slide, I'll show you the fibers in a microscopic view. Also, you can use another kind of collectors, a rotative collector that meanwhile the fiber is formed in the air. This one is collected in a rotatory collector that align the fibers, as you can see here in this GIF. Align collectors. In the upper right GIF, you can see how the electrical force attract and form the nanofibers in the air. So the solvent is evaporated in the air and is collected randomly in this case and aligned in this other uh, option. This figure uh, at the left and at the right, sorry, and the right side, you can see the comparison between electrospun nanofibers that are 1,000 times smaller than human hair. So you can see the magnification of these nanofibers. Prepare nanofibers is kind of the technique is kind of easy, but requires to play with different conditions. We had three kinds of condition. For example, we need to see and evaluate what molecular weight of the polymer is the adequate for the optimal fibers. In the electrospin technique, we want to obtain a morphology, an homogeneous morphology like this one. So we want to obtain fibers with with uh, without any imperfection, without any bulb, without any. We can prepare nanofibers that has similar fiber diameters. So for that, we need certain conditions like the molecular weight, the polymer concentration of the solution, the solvent conductivity or the polymer conductivity, the surface tension. And we play with different conditions in the electrospinning technique. For example, the applied voltage. As mentioned before, we can use 5 kilovolts or we can use 20 kilovolts depending of the polymer usage, of the conductivity of the solution. We will apply different voltage. The distance between the collector and the tip of the shrink is also important. If we have a bigger you have a bigger distance between the collector and the shrinks, you will have a thinner fibers, and if you have them closer, so you will have very thick fibers. The flow rate is also important, and the type of the collector used will determine the distribution of the fibers. Also, ambiental parameters are also important. For example, temperature and humidity will determine the formation of the fibers or not of the morphology. In the case of the antimicrobial activity of a loaded polymeric fiber with silver nanoparticles, we load these nanoparticles because these nanofibers will serve as a drug delivery system, because when this nanofiber degrades, this degradation will release silver nanoparticles in the target tissue. So silver nanoparticles are highly reported to have this antimicrobial activity, and I show you some mechanisms reported in the literature. For example, they can bruise the membrane. They can modify certain proteins in the at the surface. They can inhibit DNA replication. They can provoke the releasing of ROS, reactive species. They can react, or they can join to TIOS groups at the membrane, and so on. They can do many things that avoid the equilibrium of the cells and provoke apoptosis. The release of the fibers, as I mentioned before, the fibers serve as a drug carrier. So you can see a loader fiber here that all silver nanoparticles are encrusted between the fiber, over the fiber, and inside the fibers. And meanwhile, the fiber is degrading, they release silver nanoparticles. These technologies we want to propose it for wound healing. So we can add the membrane, the bioactive membrane into a wound, and we will protect the wound from bacterial infection, and at the same time will promote cell migration and cell proliferation at the skin in order to make faster regeneration in a wound, in a skin wound, for example. I show you more closet how a wound dressing is is done. This, for example, is a publication that we have, that we prepare a multi-layer system loaded with dexamethasone sodium phosphate to release it in the oral cavity. So you can see that the dressing, the multi-layer dressing, that is easily handly. You can easily manipulate this final dressing, and this one is loaded with some this dexamethasone drug. You can see how in the middle, in the figure that is in the middle, you can see the fibers, how they are forming. So this structure here, when you have the polymer solution that is attracted with the electrical field, and you can see the creation of the fibers, this point is named as Taylor cone, a Taylor cone. So this is another schematic representation of an ideal dressing. An ideal dressing usually protects the wound from pathogens, and also helps to absorb the exudate of the wound. Also, our polymers or system is configured to keep the environment hydrated and permit the gas exchange. These fibers of these wound dressings also will stimulate the wound healing and the epithelial bud, as you can see in this following scheme. And this work in particular that is in process right now, we are characterizing still some samples, uh, compare different methods to incorporate silver nanoparticles on the nanofibers. And why the necessity to search for this, for the best incorporation method? Because we found several methods. So it's important because silver nanoparticles usually aggregates if we blend the silver nanoparticles into the polymer solution directly. It aggregates, and this aggregation avoid the reactivity of the silver nanoparticles, and this also provokes some oxidation of the silver. So we study different methods to incorporate uh, the silver nanoparticles and avoid this aggregation or oxidation of the silver. We found in literature 4 different methods for silver nanoparticles incorporation: direct blending, UV irradiation, silver mirror reaction, and thermal reduction. In the case of the direct blending method, you previously prepared silver nanoparticles and blended to a polymer solution. You blend both, you mix both components with certain temperature, certain velocity, depending on the polymer choose, and then make the electrospinning technique. So you will obtain nanofibers like this one with encrusted silver nanoparticles. You can see some silver nanoparticles just at the surface, a little bit at the surface of the fibers, or uh, most of them almost at the inside of the fibers. This has several advantages and disadvantages. For example, between the advantages of this direct blending method is that is the easiest one, because there is only one step method, meaning that you blend nanoparticles with polymer solution and then make the electrospinning technique. Is quicker, is easier compared to the other methods. You will see the other methods. And the polymer solution itself is the reducing agent. Okay, so you can prepare the silver nanoparticles. You can add some silver nitrate directly to the polymer solution, and this polymer solution, the solvent, will help to reduce the silver to silver nanoparticles, because it's the easier one, is highly studied, and you can compare your results with literature, and you can obtain particle size about less than 10 nanometers. Between these, the disadvantages of this technique is that silver nanoparticles usually gets aggregates during the solution preparation. Uh, Sometimes the prepared nanoparticles, you need to do it before. You had to prepare silver nanoparticles before this process of electrospinning. So the dispersion of nanoparticles in the electrospinning solution is also challenging due to the high viscosity of most of the polymer solution, because to create electrospun nanofibers, you had to prepare a high viscosity solution, polymer solution. Uh, Also, uh, one of the disadvantages is that you need to protect the solution from light more than 6 hours, or the time required for the polymer dissolution in the solvent, and you have to protect the silver. You cannot control the particle size and the number of silver nanoparticles distributed on the nanofibers. And usually this technique prepares fibers with lower antimicrobial activity compared to the other methods. And that's because uh, silver nanoparticles aggregates and some of them are inside the fiber, so the bioactivity of these fibers get lower. We have another method like UV radiation method where you prepare the polymer solution, you create the electrospun fibers, then you submerge these nanofibers to silver nitrate solution, and then you expose this solution with these nanofibers inside the solution with UV irradiation. So the silver nitrate solution get reduced over the fibers, and you can obtain uh, a more distributed and with higher quantity of silver nanoparticles over the surface. Between the disadvantages of this UV radiation method is that some polymers degrades with UV. You can use a higher radiation like more than 380 nanometers. Uh, so most of the polymers get affected with UV light, but you need an extra UV irradiation step treatment for about 3 to 24 hours. This irradiation time is prolonged, so if you can control this time of irradiation, you can get uh, a higher size nanoparticles. So you need to take care about the time you will irradiate your nanofibers with the UV, because you will obtain undesired sizes of the silver nanoparticles. Between the advantages that we have with this technique is that you don't need an extra reaction time. For example, an additional solvent, because in other techniques you need to use additional solvents. So you just use the polymer, the solvent that you will dissolve the polymer, and the silver nitrate. That's it. Is also the second mass study. So this is important because you can compare your results with literature. It has better antimicrobial activity than the blending method. You can control the particle size and quantity by the irradiation time, and you can obtain very small particle size, for example, less than 10 nanometers. We have another method when instead we irradiate with UV the fibers, we can expose the electrospun fibers submerged into the silver nitrate solution with temperature. So we prepare the polymer solution, we create the nanofibers, we submerge nanofibers into the silver nitrate solution. This system you put it in a drying oven about 160 Celsius degrees for certain time, and then the silver nitrate solution get reduced, obtaining silver nanoparticles all over the surface of the nanofibers, as you can see in this same figure. This method is simple. You can get uniform distribution of nanoparticles. The size and distribution of the nanoparticles is controllable depending of the time used and the temperature applied. You can also obtain very small particle size, less than 10 nanometers. Uh, but not all polymers can support 100 above of 100 Celsius degrees. For example, some polyester or natural polymers that degrades with lower temperatures. And pues this thermal reduction is an additional step, and it can take from 1 to 12 hours extra of the just blended electrospinning. Sometimes these nanoparticles falls or leak out of the fibers because they are not very pretty stick to the fibers surface. Finally, the most complex but one of the most efficient methods for loading silver nanoparticles on fibers is the silver mirror reaction, where also you prepare the polymer solution, you prepare electrospun fibers. These fibers you submerge into the silver nitrate solution, and then you add additional solvents that will reduce this silver nitrate. You can use ammonia, you can use formaldehyde, you can use different kind of reducing agents. So also the time and the concentration of the reducing agents are important, and you can obtain a pretty coated nanofibers, as you can see in the same figure. From the advantages of this last method that we will discuss today is that the reaction can be doed in room temperature. You can coat large structures, for example, a telescope glass. You can coat also microstructures, for example, the nanofibers. You can create soft coatings, easy to control. Reducing agents may vary. So there are cheaper ones, there are more effective ones, and you can obtain also a very small particle size, less than 10 nanometers. The disadvantage for this technique is that you need a reaction volume of 25 or 30 milliliters. So you have to use a high quantity volume of the regions. Or sometimes, to eliminate the reducing agents, you need to purify or filtrate or wash or vacuum dry these final resulter membranes. And for this, you need to add 2.5 hours after the treatment of the electrospinning technique. So you need to use another kind of additional regions to stabilize the nanostructures. It's difficult to control the nanostructures, and uh, before the electrospinning technique, you need to use still 12 more hours. So this technique is the longer one. So we want to as we found many different results in literature, and all of them report different things. They characterize different. They prepare or they evaluate the antimicrobial activity in different ways. So we think that it will be useful to compare these four methods with same conditions. We had different samples here with the four different methods. Also, another thing that I didn't explain before, that uh, nanofibers For example, if I get here, nitrate solution, silver nitrate solution can be added. Another way to do this silver mirror reaction of the thermal reaction or the UV irradiation method is that you can, before the electrospinning technique, you can add the silver nitrate. You can add it in the polymer solution, creates the nanofibers, and then add the uh, the reducing agents. In the case of the thermal reduction, you can add, first of all, the silver nitrate to the polymer solution, creates the nanofibers, and then uh, prepare the treatment with temperature. Or you can add the silver nitrate to the polymer solution, create the nanofibers, and then apply the UV light. So we prepare different samples with the different alternatives of the methods. Okay. So here we have uh, uh, 7 different samples where we electrospun, or we treat the electrospun fibers before and after the electrospin. O sea, we add the silver nitrate before the electrospinning technique or after, and we compare the different results, the different conditions of the electrospinning technique. Hopefully, don't confuse this explanation to you. So we take some pictures with microscopy, same microscopy, and we compare the different loading of the different um, fibers. As you can see for silver mirror reaction uh, that all the fibers are coated with the silver nanoparticles that are the white dots on the figures. Also, we can see a pretty nice loading for the UV irradiation method in both before and after the electrospinning. You can see the small white dots through the fibers and all over the fibers. Maybe these two of the silver mirror reaction is too much. So most of the silver nanoparticles are released in a fast release. So sometimes you need, as I mentioned before, you need to release the silver nanoparticles when the nanofiber is degrading. Okay, so UV irradiation is the most promising loading uh, method when we speak in terms of morphology in these same images. We obtain fibers from 600 nanometers to 800 nanometers. So the bigger one, the thicker fibers are the control ones that has no silver nanoparticles in the fibers. So for these electrospun nanofibers, we use polycaprolactone and polyvinyl pyrrolidone polymers. Polycaprolactone is a polymer that is insoluble in water, degrades very slowly, and gives uh, very interesting mechanical properties to the wound dressing. Remember that these fibers are proposed for wound dressings, and the PVP, the polyvinyl pyrrolidone, is a polymer that absorbs water and will gives this moisture to the site when the wound dressing is located into the wound. Meanwhile, the healing of the wound. We also measure the silver nanoparticle sizes, and we see that with the thermal method, we obtain the smaller nanoparticle size, and the higher one was with UV, UV and direct blending technique. But if you compare it to literature, for example, in these other works, they they use they use the different methods. I found that 4 different methods, and they have these several silver nanoparticles. Just remember that they look for an optimized uh, time or optimized temperature, and I prepare these silver nanoparticles with same conditions. For example, in all of them, I irradiate for 1 hour with the UV light. Okay, and I I treat the nanofibers with just 1 hour with high temperature, 160 degrees, and the solution with the reducing agent was only 1 hour. So as you can see, for all of the samples, I use a similar condition in order to compare them and choose which one of the methods are the best to load silver nanoparticles with similar conditions. The below literature look or search for an optimized condition in their specific method. So that's why they obtain pretty small silver nanoparticles. But as you can see, and for UV in thermal method, we obtain very good particle sizes. We also evaluate the infrared spectroscopy. We look the specific signals of the both polymers. So most of the signals are kind of the same. So we have very low proportion of silver nanoparticles. We use 1% of silver nitrate into the preparation. So this is pretty low. So we also not see any alteration of the signals, important signals. We also evaluate the biocompatibility, or best said, cytotoxicity in HFF1 human fibroblast cells using the MTT assay. So this MTT assay evaluate the mitochondrial functionality of live cells. So 100% is the normal growth of the fibroblasts, and we expose these fibroblast cells to the different samples, and as you can see, all of them has uh, more than 80% of proliferation after 24 hours. So it can be said that these fibers, all of them are non-cytotoxic. We also expose E. coli and Staphylococcus aureus to these electrospun nanofibers, and found that, for example, fibers exposed to UV irradiation and thermal treatment has or possess the highest antimicrobial activity. So we expose E. coli to 3 days, 24, 48, and 72 hours. And we found that at 48 hours, these 4 samples uh, kills more than the 50% of the population of E. coli. In the case of Staphylococcus aureus, in the case of the Staphylococcus aureus, I'm sorry, uh, we we also obtain good results. As you can see, UV irradiation UV irradiation method and thermal irradiation method also present a lower survival of bacterial cells because as you can see here, this is the 50%. So most of them present a high antimicrobial activity against Staphylococcus aureus. We also found that a direct blending method and silver mirror reaction also possess high antimicrobial activity against Staphylococcus aureus. So right now we send our samples to get evaluated with to evaluate the thermal behavior in order to see other evidence of the silver nanoparticle presence in the nanofibers. And also we want to see if this incorporation of silver nanoparticle uh, alterate the thermal behavior of samples and provoke a certain degradability, a faster degradability when temperature increase. Also, it's important to measure mechanical properties in order because they will use as wound dressing, so it needs to be manipulated by the user. So it's important that these fibers does not breaks when the patient of the user take it and put it on the wound. Also, we want to prepare other pictures with SEM and use the EDS filter in order to see if we can find any silver signal on the fibers. And finally, we want to choose a system and prepare and evaluate some design some in vivo testing with some animal model. From the preliminary conclusions, where all methods were prepared with similar conditions and produce silver nanoparticles from around 17 to 122 nanometers. So those are pretty good sizes. The best method that we can set for producing the smaller silver nanoparticles on the fibers was the thermal treatment before the electrospinning. That was the best method that has the best distribution in on the fibers. All PCL PVP loaded with silver nanoparticle fibers show no cytotoxicity against human fibroblasts. And thermal treatment before electrospinning provoked the highest antibacterial activity in E. coli and Staphylococcus aureus. So we have already a champion. So these are only preliminary results. We're still working in several other studies. Hopefully, you are interested in this technology, and we can talk later and see we can collaborate. So please, any question, let me know. Thank you.